In this lecture, we will be discussing proteins, tertiary structure, and also quaternary structure. So if you remember from Biology 115, the tertiary structure is the 3D structure. A 3D, the overall fold. Okay. So you're going to name the forces that stabilize tertiary and quaternary structures. Compare structure and function in globular and fibrous proteins, naming specific examples. This is really important when you're given examples and you're not exactly sure what they mean. At least in this way, you will know. Compare the diversity of tertiary structures in terms of common structural motifs. So what kind of themes do we see that are really chemically based? And you also need to be able to explain the role of chaperones, such as the GRO, EL, and ES. So again, we're naming specifics because if you see this on an exam, or if you're talking to somebody and they mention this, you should know what it means. Okay, first off, there's two main classes of tertiary structure: the fibrous and the globular. So the fibrous has really defined secondary structure, and its tertiary structure, its 3D shape, is going to be long, sort of elongated, and a lot of times it could either be mostly either alpha helical or beta strands or beta sheets together. Globular, however, is less well-defined secondary structure, so it has a variety and actually a mix of alpha helices and beta strands. And of course, overall, these structures lead way to the function. So most fibrous proteins are used for structure and support, whereas most globular proteins are used as enzymes. These are used for catalysis. So globular proteins have a very defined inside and outside, whereas fibrous, it's not, they don't have a clearly defined inside portion that's accessible to water. Okay, examples of fibrous proteins include the keratins. So keratins are found pretty ubiquitously throughout your body, and these come in two different forms. The alpha keratins are mostly alpha helices. The beta keratins are mostly beta strands. Another well-known fibrous protein is collagen. So a lot of people buy lotions with collagen in it, and that's kind of silly. And we can discuss why in class. Here we go, keratins, collagen. So collagen is really interesting because it's made of mostly alpha helices, but then they're also woven together. And so pardon my sort of rudimentary drawing here. So this would be like one alpha helix strand, and then another one, so you can imagine they're like braiding together, and then a third. So by interlocking all of these different alpha helices, this makes it extremely strong. So an interesting thing with the collagen structure is it has a lot of glycines in it. So it's glycine and then something else, and then another glycine, and this unit sort of repeats in all three of these helices. And this glycine, it being so small, allows these helices to be very flexible and form together to form this triple helix structure, which are actually left-handed helices. And that's another story for another time. Okay. So on to globular proteins. So these have a variety of alpha and beta, and these are a lot of times found in enzymes. But as we've been studying biochemistry for decades now, and what a lot of biochemists and molecular biologists have noticed is that there's actually repeating patterns of alphas and betas that can be called motifs or domains in the three-dimensional structure. Okay, so The idea is that secondary structure motifs, so patterns of alpha helices and beta strands, will then make up the 3D domains. A protein can have more than one domain in it, and typically domains can be associated with a particular function. So your book has very beautiful illustrations on this, and I'm not going to try to draw it, but there's a few main themes. We have mostly alpha helices, and then we can have mostly beta strands, and then there's some that are a combination of alpha plus beta in repeatable motif. And then there are some that are just loops. It has very few secondary structures, so few alpha or beta. So one of the common domains that you're going to see is the Rossmann fold. So this is found a lot in dehydrogenases. R-O-S-S-M-A-N, yeah, just one N. It's the Rossmann fold. So this is really good for binding cofactors, and this shares an alpha-beta motif. The secondary structure repeating like this. Beta, alpha, beta, alpha. So it's got two repeating units, alpha, beta, alpha, beta, alpha. Alpha, beta, alpha, beta, alpha. Okay. But the tricky thing is, this is actually the N terminus, and this right here is the C terminus. Okay, and then this leads to this, and so on and so forth. So what this gives us, this goes up and connects here, then connects like so. So this actually gives us a pattern of parallel beta strands, as you can see. So here's where they look like in arrows. This is the 3D representation. So this would be a motif, and this whole thing would be the domain, the overall fold. Okay, and so here's what it looks like in 3D. And you can see the beta strands together make a beta sheet, and you see it's number one pleated and kind of curved. So they don't stand completely straight. And so this is a major theme in protein folding, is that these beta sheets are kind of splayed. Okay. And again, blue, this would be the N terminus, and red, this would be the C terminus, which are also labeled here. So altogether, these motifs combine to form this domain, really great at binding NAD, really great at binding ATP. There are other domains, such as the alpha-beta barrel. And so that just kind of looks like alternating alpha helices and beta strands. Let's see if I can find a nice picture of that. Okay, here you go. And this is what an alpha-beta barrel would look like in the 3D representation. So we see that what this actually does is it kind of makes a hole in the middle, like it could hold something like a barrel. So the alpha helices are around the outside and the beta strands are in the middle. And so this is actually a really great scaffold that's used by many, many enzymes to perform tons of different catalysis. So both of these, the Rossmann and the alpha-beta barrel, are obviously a mix of alpha and beta, which is quite common. Another would be the beta barrel, which, as you can imagine, would be made up of entirely beta strands. So these would be anti-parallel much of the time, and you would get a similar result to this, but the beta strands end up making a 3D shape together that looks kind of like this. And if you look from the top down, again, it makes that barrel. Same thing with alpha helices. Alpha helices can make barrels as well, all on their own. So this huge variety of structural motifs make up these different domains, which can have designated functions, or it can have varieties of functions. For example, some of the beta barrel proteins are used to drill holes into cell membranes and cause ions to leak out in the cell to die. That's common in some toxins. Okay. And it wouldn't be talking about 3D structure of protein unless we talked about how the 3D structure of proteins happens, and it's through these magical proteins called chaperones. And the most common examples of chaperones are the GRO-EL and the GRO-ES. So I'd like to point out that given enough time, proteins can and do fold on their own in physiological conditions that mimic the cell. However, we know that many chemical reactions need some help in order to occur at the pace at which cellular life processes happen. So that's really what the GRO-EL and the GRO-ES do. So as you can imagine, the structure of these really helps aid in the function. So this is 2 subunit complex, and the middle can hold up to a 60 kilodalton protein at a time. So this is really, really big. And this would of course be denatured, and what that means is unfolded. Okay. So fun fact, GRO-ES is at the top of this ring, and the 2 subunits below are the GRO-EL. Okay, so basically the protein will enter here, and this environment on the inside is perfect for the structure to form. And then this has to be in pieces so that when the protein has completely folded into its correct shape, the subunits of the chaperone can just fall apart, and they can reassemble some other time. So in order to make this process really favorable, this does require the use of ATP. Which is something that we're going to be seeing a lot of. So protein folding itself is really sort of complex. The chaperones help it along by helping the non-native, the denatured form, find its preferred energy state, which we used to think of like a well. But we realized that it's more like a series of wells. And there's like a 3D map, right? So it can start folding and get to some lower energy state, but that's not right, so it comes back out, and there's a variety of different paths to get to the lowest energy state. And this is actually a very complicated process when it happens outside of a protein chaperone. Okay. And this leads us into talking about the forces that drive the folding process to determine the tertiary structure. And a lot of this has to do with the side chains of the amino acids. So secondary structure was a lot of hydrogen bonding of the backbones. That is still true here. Hydrogen bonding is very important in tertiary structure as well, but so are hydrophobic interactions. So are ionic bonds and salt bridges. So are those disulfide bonds. All of those play a role in starting the folding process, getting it down to that lowest energy state where the hydrophobic side chains are away from water and the hydrophilic are towards the water. Stabilizing that structure using salt bridges, using disulfides, using hydrogen bonding. It's a very complicated process, which is why there's so many different, so here's the energy, right? Energy increasing this way. That's why there's so many different wells here. There's so many different paths in order to reach the lowest energy state, which would be the best state for the protein to be folded in. That's its native state. here it would be denatured.